In this lesson, we'll begin our discussion of neurophysiology. I want to talk about three different topics. The first one is the resting membrane potential. Here we're going to take a look at what a neuron looks at from a physiology point of view, what it looks like at rest when it's not firing. Then we're going to take a look at two types of nerve transmissions or nerve impulses. One is known as a graded potential, and then the other one is the actual nerve impulse known as the action potential. But before I can talk about graded or action potentials, we need to know about what a neuron looks like at rest. And this is known as the resting membrane potential. I usually abbreviate this as RMP, right? Resting membrane potential. And again, this is what a neuron is doing when it's not firing, right? While it's resting, it's not doing anything. So this is a form of potential energy. And if you remember from previous lessons, potential energy is stored energy. And what we're going to see is a buildup of negative charges inside the cell membrane, and then a buildup of positive charges outside the cell membrane. When the membrane in this case, or anything, has a separation of charges where there's positive on one side and negative on the other side, we say that structure is polar, right? And a perfect example of this would be a battery. If you took a AA battery, or even your car battery, right? If you look at the AA battery, on one side there's a positive sign, on the other side is a negative sign. Right, if you lifted up the hood of your car and you see your battery in the corner, right, there's a terminal sticking up that says positive, and then there's a terminal sticking up that's negative. Both batteries, whether it's a AA or your car battery, has stored energy. It's potential energy that we can use. And we say those batteries are polar. Okay, so that's what polar means. There's a positive charge on one side and a negative charge on the opposite side. So let's take a look at a picture and then come back to this sentence one more time. What we have here from a previous lesson, if you remember, this is the soma or the cell body, and the picture here is taking a piece of the cell membrane, and we're blowing it up here, right? So the blue is going to be the cell membrane of the cell body. And then notice on the outside, the extracellular fluid, we have all positive charges. And then on the inside here, all negative charges. So why is that? And by the way, these are protein channels, right, that are inside the cell membrane. These are actually gated channels. We'll talk about them in a minute. So why is the inside negative and why is the outside positive? Well, the reason the inside is negative, inside of our cell, we have phosphate ions, which are strongly negative charged. We have large amounts and large in size proteins that are also negatively charged. So they tend to give the inside of the cell a negative charge. Now inside we also have potassium. Even though potassium is a positive ion, these proteins and phosphates, they're so negative that they make the inside negative. What makes the outside positive are sodium ions, right? The outside of the cell is rich in sodium. Right, so now you can see we have a separation of charges, positive on the outside, negative on the inside. Let's go back to our notes for a moment. Okay, so the resting membrane potential is again a form of potential energy, stored energy like a battery, with a buildup of negative charges inside the cell and then a buildup of positive charges on the outside. Now, you know that batteries are not all the same strength, right? That AA battery has a different strength than your car battery. We can measure the strength of a battery. In neurons, the strength can range from minus 40 millivolts all the way down to minus 90 millivolts. On average, we're going to say the average resting membrane potential, and I want you to know this number, is minus 70 millivolts. So if you took a volt and you chopped it into 1,000 pieces, right? Milli is 1,000. Took 1 volt, chopped it into 1,000 pieces, and took 70 of those pieces. That would be the strength of our battery. Okay, so I definitely want you to know that number. And by the way, we have to say minus 70 millivolts. Why? Notice here, they took the voltmeter, right? They stuck the needle into the inside of the cell. The inside is negative, so we have to say negative 70 millivolts, right? On an exam, if you see a choice positive 70, right, don't pick that. That is not correct. It is negative 70. We're measuring the inside. Okay, so what are some of the factors that determine RMP? So the extracellular fluid, as I said before, is rich in sodium. That's the predominant ion out there. It has some potassium, and it also has some chloride. Even though chloride is negatively charged, there's so many sodiums and potassiums, right, predominantly sodium, that the extracellular fluid is positive. The intracellular fluid, like I said before, has large proteins which are negatively charged and phosphates which are negatively charged. And it does have some potassium, even though they're positively charged. These large proteins and phosphates are so negative that the inside is negative. Okay, so we need to make sure that that's always the case, that the outside is positive and the inside is negative. So that's what I want to talk to you about now. This is known as maintaining resting membrane potential. So here's the deal. Our neuron cell membrane is naturally permeable to sodium and potassium. And what that means is sodium can move from the outside of the cell in, and potassium can move out, right? It will allow potassium to diffuse 3 times for every 1 sodium, right? So for every 1 sodium that may leak in, 3 potassiums leak out. If we just left that alone, eventually we would have an equal number of sodium and potassiums outside, and we would lose polarity, right? Everything would be positive. It would be positive outside and positive inside, and our neurons wouldn't function. So we can't have that. So there's two mechanisms for maintaining resting membrane potential. So the first one I call like an electrical pull of the potassium that's diffused out of the intracellular fluid. So here's what happens. As potassium diffuses out of the cell, the inside of the cell is going to become more negative, right? Think about that. The inside of the cell is negative. Now we have potassium leaving. Potassium is a positively charged ion. That means the inside of the cell is going to become more negative. Well, that strong negativity actually attracts and pulls the positively charged potassium back into the cell. Remember, opposites attract, right? So that electrical pull is going to bring the potassium back into the cell where it belongs. In addition, our cell membranes of the neurons have sodium potassium pumps, and they're consistently working to pump sodium out of the cell that has leaked in and pump potassium back into the cell that has leaked out, right? These require ATP, right? Our pumps don't work because they're active transport. They don't work without ATP. Okay, so I had mentioned before, in the cell membrane, we have these proteins that are going to function as channels. So I want to talk to you about two different types of channels that are in our cell membranes. The first one is just an open channel. It's open all the time, known as a leakage channels, right? These are channels that are always open, allowing those sodiums, to move in and potassiums to move in and then out, that type of thing, or move out, then move back in. They're just kind of open. So they're like a tube or a tunnel in the protein. They're just naturally there. But we also have gated channels, right? That's what this is here. This is a gated channel. Notice the channel is closed. Right? We'll talk about in a minute how it opens up. These are known as gated channels. They're proteins, and these are channels that allow only certain ions to pass through in response to certain stimuli. And what I'm saying there is they won't open unless we stimulate the neuron. There's 3 types of stimuli that I'd like you to know. The first one is called a chemical stimulus. This would be an example of putting a neurotransmitter on the cell. like acetylcholine. Another type of stimulation or stimulus could be mechanical, right? Some physical, pressure, that type of thing, something touching the neuron. And then the third one can be an electrical stimulus, right? Any type of change of voltage, any type of electrical voltage change in the area, that could stimulate the neuron also, right? So all three of these would open these gated channels right here. Okay, so now that we know what the neuron looks like at rest, let's take a look at our first type of nerve conduction or nerve impulse known as a graded potential, right? This is what I'm going to call it, but just be aware that sometimes people do call them local potentials. These are changes in membrane potential. So we're going to see that voltmeter change from minus 70, it's going to change. And these will now spread a little bit away from the point of stimulation. Okay, they don't go very, very far. And as they go further away, they actually get weaker. They're produced by any stimulus that opens a gated channel, right? So any stimulus that opens this channel here, whether it's chemical, mechanical, or electrical, that would cause that to open. As an example, a resting membrane is exposed to a chemical, let's say acetylcholine. Here's what's going to happen, and I'll show you on a picture, and we can even go back to these notes. The chemically gated sodium channels are going to open. Right? When they open, sodium is going to come into the cell. And as the sodium enters the cell, the membrane potential rises. And what I mean by that, it's going to become more positive. So it's minus 70. As sodium ions come in, right, they're positive, the inside of the cell is going to become more positive. That we call depolarization. Now, as the sodium ions come in, they produce this local current. The sodium ions, what they're going to do is inside the cell, they're going to kind of spread laterally, and they're going to depolarize the next segment of the cell membrane. And then that, it's going to spread that way. I'll show you on a picture. So as the sodium ions spread laterally, we call that a local current. That now depolarizes nearby regions of the plasma membrane, and that's the graded potential. Um, the change in potential is proportional to the stimulus. So if you have a light or a weak stimulus, that graded potential isn't going to spread very far. If it's a stronger stimulus, it's going to be a greater graded potential. Let's take a look. So we'll begin here at the resting state one more time, just to make sure everybody's comfortable with this. Here's our cell membrane of the cell body, right? We're in the cell body here. You have your gated channels here, right? These are proteins. The charge on the inside of the cell is negative. The charge on the outside of the cell is positive. And we know it's at rest because what's the number? Minus 70. Right? So this is showing us the neuron at rest. Okay, so this time we apply a stimulus. Let's just say we're going to apply a chemical. Let's just say it's acetylcholine. So if you notice, the proteins now, that red dot, see if you guys can pick that up right there. That red dot and that red dot, these are the chemicals that have come on to the cell membrane and have attached to the receptor. Once the chemical attaches to the receptor, the protein channels open, and look what happens. Sodium floods to the inside of the cell. As sodium comes into the cell, the inside is going to become more positive. Look at our meter. We just went from minus 70 millivolts to minus 65, right? Minus 65 is more positive than minus 70. Okay, but again, the only reason these channels open is because the acetylcholine, let's say, attached to the protein receptor. Now, the next thing that's going to happen, as the sodium floods in, the sodium is going to move laterally. You can see it over here, right? It's moving laterally. That movement of the sodium within the neuron is the graded potential. That's the local current. So watch what happens. As the sodium moves to the next section of the axon is becoming more positive. I take it back, not the axon. As it moves to the next section of the cell membrane, ignore axon, that was a mistake. We're not talking about the axon, we're talking about the cell membrane of the cell body. As the sodium moves along the cell membrane of the cell body, now this section is starting to become more positive. See, here we're down to minus 65. The first section went down to minus 60, right, or up to minus 60 because it became more positive. So this local current, as the sodium is flowing, it's causing depolarization of the next section of the cell membrane. Okay, and it would just continue that way. It would just, then this section would depolarize, and then the next section would depolarize, and it would just move along that way. Right? It doesn't go very far. And that's really what a graded potential is. Now, once the stimulus stops, right, say we stop putting the chemical on, we want the cell membrane to go back to normal, right? We want to get the outside positive again and the inside negative again. That process is known as repolarization. This is when the stimulus is removed and the membrane potential goes back to normal. Now, what may happen also, listen to this, potassium ion channels may open. And in the cell, we have more potassium than outside the cell. And what will happen is when those potassium channels open, potassium will leave the inside of the cell and go to the outside of the cell, making the inside a little bit more extra negative. And that is known as hyperpolarization. It's for a short period of time. And then again, the cell membrane will go back to normal. So I just wanted to point it out that you may hear this where the inside of the cell for a short period of time becomes more negative than minus 70, all right? It may be like minus 80, let's say, or maybe even up to minus 90. And that's because of these potassium ions leaving the inside of the cell. So this is what we call hyperpolarization. All right, some characteristics of graded potentials. Um, the membrane potential is most changed at the site of stimulation, right? So that's where we tend to see most of the electrical changes that we just talked about. And as it travels away, the effect will decrease with the distance. So like I said, they don't usually go very far. The graded potential spreads passively. We don't need any energy. Right? It's just that natural movement of the sodium laterally. And as I think I said this before, a stronger stimulus will produce a greater change, right? A lesser stimulus will be a lesser change, right? So a stronger stimulus will have a greater change and again affect a larger area of the cell membrane. Right? Greater potentials often trigger specific cell functions. For example, they could do exocytosis of glandular secretion, right? So causing a gland to release its product. Right? We don't really even need a nerve impulse to do that. A graded potential is enough. When acetylcholine crossed the neuromuscular junction and stimulated the receptors on the motor end plate, that actually produced a graded potential. Right? And then that graded potential will travel along the muscle fiber. Okay, so we know resting membrane potential. We know what local currents are, a graded potential. But once we get that graded potential to travel to the axon, on the axon, we're now going to have an action potential, which is the nerve impulse. So this is going to be a little bit more involved than what we saw with the graded potential. These are propagated changes, and what I mean by that is that these are changes that are going to travel down the axon, and they affect an entire excitable membrane, right? So this is going to affect the axolemma, which is the cell membrane of the axon. And it's going to begin at the initial segment. So again, from a previous lesson, we had said that the axolemma is the cell membrane of the axon, and just after the axon hillock is the initial segment, and that's going to be the first place that the action potential starts. Now, unlike a graded potential, as the action potential moves down the axon, it doesn't get weaker. It stays the same strength, right? So they don't diminish. These are stimulated by graded potentials that depolarize the axolemma to threshold. So all I'm saying as the graded potential comes, say, to the, let's go back a moment here, right? See how the graded potential would travel along the cell membrane of the cell body, and then once we get to here, the initial segment, that will now trigger an action potential, which would travel down the axon. And so that's all I'm saying. Um, and the threshold, so I'm going to talk about this next. So the threshold of an axon, meaning that once we get the inside of the cell at the initial segment to about minus 60 or minus 55 millivolts, once it becomes that positive, the action potential will be initiated. The number that I usually ask on an exam, the number that I want you to know is minus 55. It does range from minus 60 to minus 55, but let's focus on this number. If I were to ask you what's the threshold value, it's going to be minus 55. And again, I'm going to talk about more about that in a minute. So let's take a look at what an action potential is. Let me read the definition. It's not going to make a lot of sense. We'll look at a graph and then we'll maybe go back to this definition, see if it makes more sense. An action potential is a sequence of rapidly occurring events that are going to decrease, eventually reverse the membrane potential, and then restore it back to normal. All right, so we're going to see some changes from our resting membrane potential. We're going to divide the nerve impulse into two parts, depolarization and repolarization, right? We just saw these words in graded potentials. We're going to use them again. So I'm going to skip the notes and let's take you to a graph. So let's just say we were in a physiology lab and we had a neuron hanging on a hook and we attached some recording equipment on it to measure the voltage changes inside the axon. So as we would be reading the voltage changes, let's start here. Would everybody agree that in blue, this line here, this would represent RMP, which is minus 70. This is the neuron at rest. Let's just say right here, I stimulate the neuron. Let's say I take maybe a little electrical stimulus and I touch the neuron, or I put a chemical on it, like acetylcholine. What'll happen is this. Some sodium will start to flow inside the cell. Would everybody agree? As sodium flows in, it produces a graded potential. If enough sodium comes in that we reach threshold, and again, the number that I want you to know is minus 55. Here they put minus 60, which is fine because it is a range, minus 60 to minus 55. But again, on an exam, I usually say minus 55. If the inside becomes positive enough that we get to minus 55, at that point, sodium gated channels open up in mass majorly, and tens of thousands of sodium ions flood to the inside of the cell. As sodium floods to the inside of the cell, the curve quickly jumps up like this, all the way to plus 30. This upward swing here is the depolarization. Why do we say depolarization? So remember earlier on, I said when something is polar, it has a negative charge on one side and a positive charge on the other side. Well, once we get to 0 and up here, the outside is positive and the inside is positive. We're no longer polar. We've depolarized, right? So this upward swing is the first half of the action potential. So sodium will flood in until the inside of the axon becomes plus 30. At that point, the sodium-gated channels close, and now potassium-gated channels open. Since there's more potassium inside the cell than outside the cell, potassium now floods out of the cell. As potassium leaves, the inside of the cell becomes more negative again. And look at this. We get a downward slope. That's the negative reading. And at some point, the inside is negative and the outside is positive. We have reestablished polarity. That's why they call this repolarization. So this downward swing is repolarization. Okay. Now notice here is minus 70. This would be RMP. Notice, like I said before, sometimes extra potassium leaves the inside of the cell and we become a little bit more negative. This could be like minus 90. They don't have it here, but it could be like minus 85, minus 90. That's from the extra potassium leaving. And that would be like the hyperpolarization, right, where we become more negative. But eventually everything goes back to normal and we're back to RMP. Okay, so the two phases of the action potential that I want you to know are depolarization and repolarization. Let's go back to the notes now. So these notes are going to explain what we just went over. All right, so depolarization is that upward swing of the action potential. Again, it's caused by the opening of the sodium gate. So once we reach threshold, the sodium gates open up in mass. And like I said, tens of thousands of sodium ions flood the inside of the cell. And the reading on the inside of the cell is going to become more positive. Once we get to plus 30, right? Once the ICF becomes plus 30, the sodium gated channels close. No more sodium can come in. And at that point, potassium gated channels open. Right, so you can see here, potassium gated channels open, causing the potassium to move out of the ICF. Why? There's more potassium inside the cell than there is outside of the cell. So things are going to diffuse from an area of high concentration to low concentration. As the ICF leaves again, I take it back, as the potassium leaves the ICF, the inside becomes more negative again, right? And repolarization is the downward swing. Just real quick here, if we were to stimulate the neuron while it's in depolarization and this part of repolarization, we would not be able to get another action potential. This is known as absolute refractory period. But if we were to stimulate it, as we get near the end of repolarization, like from this point on, to when we get back to RMP, this we could get some a new action potential generated. So they call this period of time, say from here to here, right, all along this, the relative refractory period where we could possibly get another action potential. Okay, I talked about hyperpolarization already. Again, that was this downward swing over here, right? Hyperpolarization occurs in certain neurons when potassium channels are open, allowing for like extra potassium to diffuse out of the cell, right? So now the inside of the cell becomes more negative. Now, when this neuron is in this state down here, it's much harder to stimulate. If we were to stimulate it, right, it's further away from threshold. So it's actually much more difficult to stimulate a neuron that's hyperpolarized, right? If something were to keep this neuron in a hyperpolarized state, like certain neurotransmitters, this is a little bit advanced. If we kept a neuron in a hyperpolarized state, it would be very difficult to get it to fire because it's a further away from threshold. Refractory period, I had mentioned before, that's that short period of time where a neuron cannot generate a new action potential. I didn't put it in the notes here, but there are two types of refractory period. Again, the absolute refractory period is if we stimulate this neuron while it's firing, while it's in depolarization and the majority of repolarization. It's not going to happen. We're not going to get a new action potential. But if we stimulate it perhaps, at this point, the near the end of repolarization, anywhere along here, then we could get another action potential. Now, we assumed when let's say, stimulated the neuron here, that the stimulus was strong enough for us to get to threshold. That's not always the case. Sometimes you could have a weak stimulus, and that weak stimulus would only then produce a graded potential if we didn't get to threshold. So sometimes what has to happen, we have to have, say we have one weak stimulus, we may need another stimulus right after it and another stimulus right after it to get enough sodium in to reach threshold, right? These would be called sub threshold stimuli. These are a weak stimulus leading to the opening of a few sodium ion channels. The change in potential is only slight, not enough to generate an impulse in a neuron. So again, we would probably need a few. Let's say here, let's just say we hit a weak stimulus here, and the curve goes up a little bit. If we don't hit it again, we would just get a graded potential, right? So we would have to hit like maybe multiple stimuli, multiple sub-threshold stimuli to get enough sodium in that we get to threshold. Okay. Again, that on my first scenario, we gave the stimulus and we said it was strong enough to reach threshold. That would have been a threshold stimulus, right? That's a stimulus that will open that critical number of sodium channels to cause depolarization, right? Once we get to threshold, you can't stop the action potential. It's gonna take off. All right, so the value again that I want you to know for threshold is minus 55, right? It does range from minus 60 to minus 55, but we're going to go with minus 55. Again, once threshold is reached, once we get to this point right here, right, of minus 60 or minus 55, this is going to happen. We can't stop it, right? You're just not going to be able to stop it. All right, so once threshold is reached, the action potential will be initiated and you can't stop it. Last concept for this. Let's say we're in the lab and we have the neuron hooked there and we say, all right, this is great. We got a nice action potential. Let's give it a stronger stimulus, right? Let's touch it with a stronger electrical impulse and see if we get a stronger action potential. And you want to know something? You would get the same exact curve that you had the first time. And the reason is, once you get to threshold, that neuron is always going to give you 100%. A stronger stimulus does not give us a stronger action potential. This is known as the all or none principle. If a stimulus is strong enough to generate an action potential, the action potential will be generated to its fullest extent, right? So all I'm saying is if we get to threshold, that neuron's going to give us everything we have, right? So, and look at this, I put it in big words here, big letters. A stronger stimulus will not elicit a stronger action potential. If anything, a stronger stimulus may generate more action potentials, but they won't be any stronger. Okay, and that is known as the all or none principle. Okay, so in the next lesson, we'll take a look at how the action potential travels along the axon.